Thank you for joining today's webinar about the PROSPER trial, a new phase 2 clinical trial for progressive supranuclear palsy. My name is Jennifer Brummett. I'm CurePSP's Associate Director of Scientific Affairs and Partnerships, and I'm moderating today's event. Before we get started, I want to share a few housekeeping notes for our attendees. Today's event will last about 1 hour. There will first be a presentation, followed by time for questions and answers at the end. There is a comments tab where you can enter any questions and answers you might want Dr. Hoglinger to address at the end of the event. And last, the webinar is being recorded, and a recording will be sent out to all registrants afterwards. Now to introduce today's speaker, Dr. Günter Höglinger, who is the Director of Department of Neurology at the Ludwig-Maximilians-Universität and also a scientist at the German Center for Neurodegenerative Diseases in Munich, Germany. His scientific work focuses on tauopathies and synucleinopathies with a translational perspective, including genetics, biomarker discovery to therapeutic trials. We're also delighted to have him as a member of CurePSP's Scientific Advisory Board. And without further ado, I will turn it over to Dr. Höglinger.
Yeah, thank you, Jennifer, for the kind introduction. Dear ladies and gentlemen, dear patients, dear caregiver, dear doctors, dear healthcare professionals, whoever is interested in this webinar, most welcome, and I'm pleased to have you here. My name, as indicated, is Günter Höglinger. I'm the head of neurology at Ludwig-Maximilians-Universität, Department of Neurology in Munich, Germany. I have the pleasure now to introduce you to a new study that is being conducted in patients living with PSP. It's called the PROSPER study. This study will be introduced in the future slides. Let me start by making some disclosures. This webinar is intended for informational purposes only, and it is designed to share details about the PROSPER study and current research efforts for treating patients living with PSP. All information presented is in accordance with ethical and regulatory guidelines, and no claims are being made regarding the efficacy or safety of any interventional treatments outside of what has been approved for discussion. The study drug is called FNP 223. It's under investigation, and the content shared today reflects what is currently known from approved studies. This webinar is not intended to offer any medical advice or to promote any unapproved uses of medications. Participants should consult with their healthcare providers for any medical concerns or decisions. Now, let me start by discussing why clinical research is of importance. You know that for PSP, we have only symptomatic treatment attempts and no disease-modifying therapy approved so far. Clinical research studies, also called clinical trials, look for ways to treat to better treat a disease or a medical condition. The study team of a clinical trial carefully monitors all risks and potential benefits of a study drug, keeping the well-being of each participant as the primary focus of research. Clinical trials follow certain rules to protect the rights, safety, well-being, and privacy of participants. The study results help regulatory agencies such as the FDA or European medical agencies to decide if a medication or product is safe and efficacious and should be available to patients. Clinical studies are essential for developing new medical treatments to enhance patient care. What are clinical trials? Clinical trials are research studies to test whether a type of intervention is effective or not. They usually test medications, but can also study new ways to prevent, detect, or diagnose diseases. In a classical conventional clinical trial setting, the recruited patients undergo first a screening period, that is a period to check whether a patient is eligible to participate in the study based on the inclusion and exclusion criteria. Then patients who are suitable are randomized, meaning that they can be assigned to either treatment with a proper drug or a placebo. Placebo is an ineffective, harmless pill, like a sugar pill, that visually looks like the study drug. It helps compare the effect of the drug to a harmless but ineffective substance as a control. Now, clinical trials can be a very, very lengthy process. They usually start with many years of preclinical work in cell cultures, model systems, which mimic the disease of interest. Then usually a phase 1 trial, which is intended to test the safety of the interventions being conducted first in healthy subjects. In a phase 2 trial, safety and dosing of the appropriate drug regimen is first done in individual patients. And then a phase 3 trial is aimed to provide evidence of clinical efficacy and safety in larger numbers of patients. These phase 3 studies usually, when successful, lead to the approval of drug, and even after the approval, after the market introduction, phase 4 studies might be continued in very large numbers of patients to generate further data on safety and efficacy. Now, you see these processes can last many, many years and consume a lot of money in order to achieve the goal. However, efforts are being made to accelerate these timings in the case of rare diseases such as PSP. For example, regulatory authorities have resources to approve an orphan drug intended for the use in rare diseases with phase 2 trials if they are positive. Now, a few words about progressive supranuclear palsy, the disease abbreviated as PSP. PSP is a rare and progressive brain disorder that affects movement and balance foremost. PSP leads to problems with postural stability and falls, gait, limb and eye movements, speech and swallowing. But PSP can also cause changes in mood, cognition and behavior. Current treatments of PSP focus on managing symptoms, but do not slow the progression of the disease. In order to achieve a intervention, a therapeutic intervention, which aims to slow the progression, we need to understand the causes and the molecular mechanisms that lead to PSP. In the case of PSP, we know that the microtubule-associated protein tau is aggregating in neurons, in astrocytes, and in oligodendrocytes, and this is believed to be the primary cause of the neuronal degeneration and dysfunction. The question is, what causes the aggregation of tau? Now, post-translational modifications, that is, modifications after the protein has been produced with small molecular side chains, such as phosphoepitopes or glucose molecules, can affect the protein's propensity to detach from the molecular targets, the microtubules, and to form insoluble aggregates, which then harm the cells. Such post-translational modifications can be either beneficial in the case of glucosylation, for example, or harmful in the case of phosphorylation. Now, OGA inhibitors are a group of new molecules which are intended to stabilize a certain form of post-translational modification in order to prevent the aggregation of tau proteins. FNP 223 is a specific form of an OGA inhibitor that targets the tau proteins and is currently in development for PSP. On the top schematic drawing, you see in the green lines schematic representations of the tau protein in the normal form, soluble form. The protein is natively unfolded, detached to microtubule, which are the railways of axonal transport in the brain nerve cells. And if hyperphosphorylated, as shown on the right side, then tau can aggregate. And if these phosphoepitopes are being replaced by O-glucNAc residues, then the molecule is kept in an insoluble manner. And this process is controlled by a protein called OGA, which is then inhibited by FNP 223. In other words, based on these conceptual theoretical considerations, FNP is believed to prevent the aggregation of tau protein. Now, there have been a number of experimental studies to test the efficacy of that intervention in models of tauopathies. That means not human systems, but experimental systems. On the left panel, you see that the treatment with the drug of interest, FNP 223, can indeed reduce the insoluble tau aggregates and increase the O-gluconatylated form of tau. In the middle panel, you see that it can, in transgenic mice, reduce tau aggregates, and in the right panel, you see that this in turn leads to a longer life expectancy of these mice. Now, the PROSPER study is intended to be a phase 2 clinical trial that will evaluate how safe and effective the study medicine, FNP, may be in treating living patients with PSP. As I have already shown to you, there is preclinical data which are supportive of its use in PSP patients. There have been phase 1 studies to suggest the safety of this intervention in healthy volunteers and patients. And now, today, we will discuss the phase 2 PROSPER study, which is intended to evaluate safety and efficacy in patients living with PSP. The PROSPER study is a study which relies on a 40-day screening period in order to identify patients who meet the inclusion and exclusion criteria. And then these patients who are deemed to be suitable for the treatment will undergo a treatment of 52 weeks. And after the treatment period, the patients will be followed up for further 4 weeks. During the participation in the PROSPER trial, participants will involve a total of 7 visits to the clinic and 7 phone visits. In total, treatment and the observation period will be 15 months per participant. Now, who can participate in the PROSPER study? You might be qualified to participate if you are aged between 50 and 80 years of age, if you have been told by your treating neurologist that you have a diagnosis of possible or probable PSP, mainly the Richardson syndrome subtype. And if you started experiencing symptoms only within the last 3 years longer, disease durations are not eligible for the treatment study because the study aims to recruit patients in their particular early course of their disease. It is also required to be able to walk independently or with a cane for at least 10 steps. And participants need to have a reliable caregiver or study partner who can accompany them to the study visits. Now, if patients are eligible and enrolled into the clinical trials, as being told, this is a randomized controlled clinical trial, they have a 50% chance to be assigned to either the active treatment, the drug of interest, or placebo. The drug, as I have already introduced, aims to reduce the abnormal accumulation of the tau protein in the brain, and the accumulation is believed to play a role in the progression of the disease. The drug or the placebo are being taken orally by mouth 3 times a day, and it's recommended to take the drug every 8 hours. That means in the morning, midday, or evening, with or without food. After the end of the treatment period, participants will be asked to attend 1 follow-up phone visit about 4 weeks after their last dose of medication in order to get a feeling about the patient's condition, safety, and possible side effects that might have emerged after the end of the treatment. So why would one want to take part in the trial? Although you may not directly benefit from taking part in the PROSPER study yourself, taking part will help us and the community to learn about FNP 223 and could help others with PSP in the future. If you qualify and you decide to take part, you will receive the drug or placebo. You can have a close medical care and follow up through the entire study course, and you're being reimbursed for the study-related expenses. Choosing whether or not to take part in the study, completely up to you. Deciding not to take part will not impact your current medical care now or in the future. And if you decide to take part, you can decide to leave the study at any time and without giving any reason for your decision. The study will will recruit 220 people living with PSP in 46 clinical trial sites in the US, in the UK, and in 7 countries within the European Union. The study is currently recruiting in the US with additional European countries now currently being activated. And the status of the activation of the sites in the region where you are living can be monitored at the website, which is indicated underneath here. There is also a website which provides further information about the trial, which is online already right now. It is written in lay language, so everybody can get the relevant information. And the website is psptrialfairer.com. So please don't hesitate to check out more information about the process study on that website. So this is the information that I wished to share with you, and I'm pleased to take your questions and to answer your questions right now.
Thank you so much for the presentation. We've had a few questions that came in during registration, and there are a few in the chat box. If anyone else has additional questions, please feel free to enter them. The first question that we received a few times is, we're wondering if you could discuss the logistics of participating in the trial. One question in particular was whether repeated travel is required to the trial site.
Yes, that is needed to participate. So I'm just checking the participants during the entire trial have to be at site at least 7 times, probably more so during the screening period. And after the trial, there is so it's 7 visits to the clinic, 7 phone calls, and probably during the screening visit, check whether or not you fulfill the inclusion and exclusion criteria, there might be additional travels. But again, as already being said, all study-related costs are being reimbursed, and that will also cover the travel cost for you and also for the caregiver, of course.
Great, thank you. We received a lot of questions about the eligibility criteria, and I know you already reviewed those. I'm wondering if you could revisit what the eligibility criteria are as a reminder and discuss the age limit for participating in the trial.
Yeah, age limit is 50 to 80 years of age. So this is probably pretty strict. If you're just 1 week younger or older, you might want to ask your treating neurologist still. Maybe there might be some exceptions. Also, I doubt that, but in principle, the rules of the trial protocol are pretty strict. So the age limit of 50 and 80. Then you need to have a caregiver or a study partner who can accompany you to the study visits and also provide information about your health status during the course of the study. You need to have a diagnosis of PSP, possible or probably of the Richardson syndrome types, and you need to have a pretty short disease duration, maximum 3 years after symptom onset, because the study wishes to recruit patients in the pretty early phase of the disease. And walking ability is also a prerequisite. So you can use a cane, but you have to be able to walk at least 10 steps independently with or without a cane.
Thank you. We also received another question asking why this trial is only for patients with PSP Richardson subtype and not other subtypes. I'm wondering if you could discuss why that is the case.
Yeah, this is a general limitation to the clinical trial concept currently and not to this particular drug. In principle, we believe if the drug is effective, it might be efficacious for all clinical manifestations of PSP because it hits the nail on the mechanism, the underlying molecular mechanisms, and not one specific clinical manifestation. The reason for including only PSP Richardson syndrome is basically because we know the natural history the evolution over time of PSP Richardson's syndrome much better than the other non-Richardson syndrome phenotypes. And in order to plan and conduct the trial in a decent manner and predict how many patients we require in order to demonstrate efficacy, clinical efficacy of the drug, we just need to rely on a phenotype that we know very well. This is the only reason. Once approved, then we hope that we can also generate additional data for the non-Richardson phenotypes. But we would expect actually that the drug would not only be efficacious in Richardson syndrome.
Thank you for that information. We received a question in the chat box asking if this medication is something that can be crushed and mixed up for patients that can't swallow.
Oh, unfortunately, I cannot answer that question. I wouldn't know. As far as I just cannot make any statement with certainty. I'm sorry about that.
No worries. I don't know is a perfectly fine answer. And if we do get an answer to it, we'll try to circulate that to folks that might need to know that. The next question we received is, since this medication is lowering accumulation of tau, do you think it might be effective, or will it be tested on other tauopathies like CBD in the future?
Yeah, the molecular mechanism of action is perfectly suited also for CBD, but whether or not it will actually work in this disease needs to be shown in the future, and whether the company will test it in CBD is up to their discretion, of course. I would think that it works for PSP. There is a high likeliness that it would also work for CBD. It requires to be shown, of course.
Thank you. We received, I actually did just receive an answer as to whether the pill can be crushed up, and it cannot be crushed up. It's not able to be so. So hopefully that answers the question around that issue. We did receive another question just commenting on the fact that this drug blocks an enzyme that's important for other normal functions. And so this person's wondering what potential side effects might be of this drug, or how those will be tested and tracked throughout the trial.
Yeah, throughout the trial, the purpose of this trial is to generate data about efficacy, so whether the drug helps to slow down the progression, at the same time also about safety. So far, there have only been limited numbers of patients human beings being treated with the drug, and the first indication looks like it's a pretty safe drug. But of course, this was based on a limited number of observations. So we will learn during the course of this trial only what potential side effects might occur. This is a matter of study.
Thank you. Yeah, it's a really important part of the clinical trial process. So it's great to hear that perspective. There's a few comments in the chat commenting on the eligibility criteria and some frustration around the fact that diagnosis can take a long time for some people. And so by the time they receive a diagnosis, it's tough to be eligible for trials. So this is unfortunately a very common frustration for patients in our community. I'm wondering if you could speak as to why the eligibility criteria are set that way, in particular around the time from diagnosis to trial participation.
That decision has been taken because for clinical trial purposes, in order to be able to have a high likeliness of really succeeding in the trial, meaning show clinical efficacy, we have the tendency to think that the highest likeliness of being successful for the drug is if one starts treatment very early. That would not exclude possibly patients once treatment is approved in later stages also receive treatment. Also, I cannot make any promises in that regard, of course, but just for having a high likeliness of being successful for the drug in the context of this clinical trial, one wants to start in a disease course which is very, very early with as low tau burden in the brain as possible. This is main reason why it's chosen, and I can understand the frustration that goes along, especially if one has a long odyssey and many months and years before the first doctor actually makes a diagnosis. But I think we cannot really change that pathway forward. Anyway, it has been decided to be that limit for the time being for this trial, and so I think once the trial is successful, the situation might look quite different then.
Thank you. One last question. We just received some questions asking if there are any medications that patients can't take during this trial. Like, for example, are they limited from taking other medications in order to participate in this trial?
Yeah, there is a list of medications as in any clinical study that are not suitable for the clinical trial. I think I cannot go through all of these. I think first it's important for you to know the list of features that I indicated, whether or not you seem to be eligible for the clinical trial. And then once you consider yourself as being eligible, please walk out to the website that I have indicated, identify whether there is a suitable trial site nearby, and then the treating doctors who care for the clinical trial will go with you in a detailed list, actually, the pros or the inclusion and exclusion criteria, and check on a very individual manner whether medications you are taking compatible. Usually, I would have the tendency that there is always a possibility to change medications once you're generally eligible for the trial, make them compatible with your participation in the trial.
Thank you. Thank you for answering all of the questions, and thank you for the great presentation. With that, we will conclude today's event. So once again, thanks to all of you for attending, and thanks to Dr. Höglinger for his great presentation. As a reminder, this presentation was recorded and will be sent out to all registrants after the event. The link to the trial website was put into the chat, and on the slides, it is psptrialforrare.com. That website will have more information on how to locate a trial center and eligibility criteria and other information for you to review before considering participating. If you have any other questions about the trial or today's event, you can contact us at info@CurePSP.org. We also hope that you'll check out our website and our social media for future events. We do several community webinars a year, and we hope to see you at a future event. Thank you all once again.